We work under the hypothesis that many forms of solid tumors grow in a hierarchical fashion, which is to say that at the apex, there is a so-called cancer stem cell that is responsible for the propagation of the tumor, would be then, by definition, responsible for reemergence of the tumor following chemo and radiotherapy. So we believe that it's identifying and isolating this cell that is most important at targeting the real tumor, because in fact most of the current therapies are aimed at dealing with cells that proliferate, and they also kill normal cells that proliferate. Here, what we believe is that in fact the proliferating cells are actually the byproduct of the cancer stem cell, and that the cancer stem cell is really the important one that we must understand and target effectively. Well, the interesting concept here is that in fact a corollary of our hypothesis is that in fact most cells in an organ are not capable of giving rise to a tumor, but that there are exactly specific constraints on the type of cell that it can actually give origin to a tumor. We refer to that as the cell of origin, the cancer or tumor cell of origin, and we believe that in most cases this is either an organ stem cell, an adult organ stem cell, or its immediate progenitor. And at least experimentally, using mouse models, which is what we do, we've shown that in fact in the brain, only stem cells and progenitors can give rise to a brain tumor called glioblastoma, and that in fact the more differentiated cells in the brain are incapable of giving rise to these tumors. So this adheres to our hypothesis, and now we've devised tools, at least in mouse models, where we can prospectively identify the cell of origin, and we can also identify the cancer stem cell. And so our hope is that through the isolation and study of these cells, we can then translate our information to human glioblastoma and to patients. Sure. So neurofibromatosis type 1 is the most prevalent genetically inherited disease of the nervous system. People with NF1, as it's commonly called, can suffer a panoply of different kinds of pathologies, ranging from intellectual deficits to autism to structural abnormalities in their bones, but most importantly and most prevalent is presence of tumors in their nervous system, in both the central nervous system, brain and spinal cord, but also in the peripheral nervous system. So they suffer from tumors called neurofibromas and other tumors called malignant peripheral nerve sheath tumors, both of which are completely recalcitrant to all known cancer therapies. And so what we've been able to do is to successfully model these tumors by virtue of exactly the same mutations in human patients, using our genetically engineered mice. And we've devised strategies to better understand how these tumors arise, what their constraints are, and do they have cancer stem cells? And happily, the answer to the latter question is yes, and we're now able to identify these cancer stem cells. So I've put together leaders in the field of neurofibromatosis for the symposium tomorrow, and they're going to be discussing the most recent advances in terms of therapies for patients, the very first therapies that are actually making a difference in the clinic. Yeah, so it's an amalgamation of a rather new idea about how solid tumors grow. I would say some would call them still controversial ideas, but we feel that we are making experimental headway in really demonstrating that this is an important new way of looking at how tumors grow, and at realizing that there is a quiescent component to the tumor, which is the one that we really must target. Yes, I think that by virtue of isolating the properties of the cancer stem cells, there will be new therapies that will either unmask them and cause them to enter the cell cycle so that they become responsive to therapy, or alternatively, completely novel therapies that target the stem cells and not normal cells. Well, I would just say that this is an extremely exciting time to be a cancer researcher, that with the coming together of the wealth of genomic information coupled with the biology, and I can't emphasize that enough, the biology of the tumors where they come from, how they grow, and their natural history is a fundamentally important component of cancer biology that is not trumped by knowing the panoply of mutations in a tumor. And so I think that I would caution the young researchers in the field to never forget about the biology, the cell of origin, and the natural history of a tumor.